All right, so like Joachim referred to earlier, I've worked a lot before on synonymous codon usage and synonymous mutations and their fitness effects. We've also done a lot of work with adaptation in beetle populations. But most of that is published, and so I want to talk about some new work that we have. So yeah, I'm happy to get feedback on this. This is not directly related to population genetics, but I think it sort of impacts a lot of population genetic models which depend on trade-offs and look at the impacts of pleiotropy in general on evolutionary effects or adaptive effects. So I'm going to talk about the incidence and impact of trade-offs. I think many of you are aware that trade-offs are ubiquitous. So for example, you have a weekend free, you can either binge watch Star Trek or Star Wars, or you can catch up on sleep, but you can't do both. Right? And so it's a fundamental trade-off all of us understand, and it's sort of true. Like, it exists, it is ubiquitous at some level. And so I'm showing you here is an example from this nice review. There's two traits. For example, you can have multiplication or you can have survival. And you either have rapid growth and no resistance, or you have high resistance and no growth and resistance to an antibiotic, for example, in E. coli. And the question is, how far can we get from these, can we get an understanding of evolution using these basic ideas about trade-offs and how ubiquitous these trade-offs are. This is a sort of summary picture from the same review talking about how many different sets of trade-offs people have figured out in bacteria. You're aware of some common ones such as swarming and biofilm formation. There's vegetative genes or resistance genes that can be transcribed to different levels. You can have antibiotic exclusion or nutrient excess and so on and so forth. Right? So for example, mechanistically, the same sets of proteins or molecules can be involved in doing different functions, and perhaps that's where trade-offs arise from in some level. So what I want to talk today about is that trade-offs are thought to constrain divergence, for example. So if you've gotten really good at some particular resource, for instance, perhaps if you're also gotten bad at another resource, in future, if you're going to see this resource number 2, you're going to have a hard time perhaps adapting to it, right, or surviving in that resource. And so in general, there's a lot of work until now showing negative trait correlations across species when it comes to particular functional traits. And this is a classical hallmark of trade-offs in a way. But intraspecific trait correlations have been relatively rarely investigated. And the reason that this is interesting and important is we don't therefore understand whether trade-offs play a major role in the early stages of divergence. So as populations begin to diverge and differentiate potentially because they're specializing on different habitats or different environments, is it true that during these early stages, trade-offs can constrain subsequent divergence of these populations and gene flow? So I want to first tell you about some work we've done with tribolium flower beetles to address this question. We collected about 20 beetle populations. This is from across India. And for about 5 to 10 generations in the lab, we allowed them to sort of stabilize. We built up large stocks. Everybody got to live in wheat flour, which is what most of these populations are well adapted to. And then we measured a large, same species, correct, yeah. And we measured a large number of different traits, which we think are important for survival and have some sort of bearing on fitness, right? So let me walk you through some of these. Here's external immunity. So beetles secrete these toxic compounds in the flour, and they are antibacterial compounds. So we measured how much of these compounds they're producing for males and for females. We measured how long they could live without under starvation. So again, for males and females. We measured the degree of larval cannibalism, intraspecific competition. So asking if they are in competition with their own population, what is the probability of a particular egg surviving, right? Similarly, interspecific competition. They're often found in close competition with a congener, Tribolium confusum. So we asked if they're competing with Tribolium confusum eggs, how well do they perform? We measure development rate, total offspring per female, adult offspring per female, and reproductive efficiency, which is just a ratio of these two. So what I want you to get out of this is that our naive expectation was we would find trade-offs, right? There should be, these are very important traits. One would expect that if there's a high investment in reproduction, you might find low investment in immune function, survival, starvation, something. And what you see largely is either there's no correlation. So each data point here is one population, right? So there's 20 populations in each of these plots. You find largely there are no correlations, and when there are correlations, they're positive. So here's an example here. Maybe this one is more interesting. So starvation lifespan is very strongly correlated between males and females of the same population. And in some sense, that's not really surprising, right? You expect this is a trait that both sexes should be under fairly strong selection for in populations, and that should evolve together in both sexes potentially. Now, the only negative correlations we find involve external immunity. So there's a negative correlation with interspecific competition. So populations that are better at producing more antibiotic compounds in the flower are less able to compete with this other species with which they're found. And similarly, reproductive efficiency is negatively correlated, but poorly so with external immunity. Right? So overall, surprisingly, we don't find any signs of trade-offs across these diverging populations. And what's important to note is that a simple explanation for this might be there isn't enough divergence, right, in these populations. Maybe these populations have, they're largely still essentially the same large panmictic population. And that's unlikely to be true because there's significant divergence because we have multiple individuals measured for each population. We know there's significant divergence, right? And so you can also see this if you look at the y-axis for each of these traits, there's quite a bit of variation. And so here's another way to look at it, is to ask, if I take the ratio of a measure of population divergence, so what is, for example, the average difference between the means of any two pairs of populations for a trait, relative to the intra-population variation, relative to the degree of individual variability we find in each population on average, that can tell us something about which traits are diverging fast, right, compared to population variation. And again, you find that immune function traits seem to be very, very fast diverging compared to variation within populations, and development rate here. And all of these traits are actually not diverging very much, right, across populations. But there is still significant divergence. Every single trait is significantly divergent across populations.
Genetically, within the population,
we don't have any genetic data on these populations yet. We have some sequencing information, but not sufficient. Yeah. Any other questions so far? Yeah, so this was sort of, well, there's no trade-offs, what do we do next? But actually it's interesting, right? Because we expected trade-offs, and there aren't many. And we also look for trade-offs across different alternate resources. So these beetles are able to eat many different flowers. There's a sampling of different flowers that are commonly found in India, for example, in warehouses. So there's Bengal gram, which is basin, jawar, wheat, finger millet, and so on and so forth. And again, you find very, very strong positive correlations here in every case. So there doesn't seem to be any selection for specific populations to specialize on different resources, right? Which is again a bit surprising because you'd expect, for example, in different parts of the country, different warehouses, some resources will be very abundant and they should specialize on these, but they don't seem to have done that. So you either have sort of super specialists who are good at everything, or you have sort of terrible specialists who are good at nothing, or relatively good at nothing. And so essentially what we're seeing then is that there are generally no trade-offs for fitness, despite substantial divergence across populations. So we suggest that therefore trade-offs may not impose large constraints during early divergence, with the exception perhaps of immune traits, and this is not surprising. Other people have also found that immune traits across various populations, various animals, are amongst the fastest evolving traits. So perhaps that makes more sense. And there seems to be quantitative variation across better versus poor generalists, but little evidence for resource specialization, right? So this is work with beetles and sort of looking at whole organism perspective, understanding a bit more about behavior of these beetles. I want to switch now to asking another question, which is, are trade-offs really ubiquitous, right? So one of the assumptions that went into this giant study was that we should find trade-offs and we would understand more about population ecology and evolution if we understand trade-offs better. And so we wanted to ask specifically about one particular mechanism of why trade-offs may occur. One mechanism is antagonistic pleiotropy. And what that means is a single mutation may improve function with respect to one trait and decrease function with respect to another trait. Right? And so at the very core, that might be one function that might lead to trade-offs. Other possible reasons for finding trade-offs are mutation accumulation or divergence selection. So for example, you accumulate lots of mutations under strong selection for one trait or one environment. And as a result, because there's weak selection on other traits, right, you accumulate a lot of mutations in other traits. And because we expect most mutations to be deleterious in most populations, all of those are going to end up being deleterious for some trait or the other. Which means now if I remove selection on this one trait, and I ask, has the organism evolved to have poor fitness on other environments, you will find often that they do have poor fitness, right? So it appears as a trade-off. I mean, it is a trade-off in some sense, but there's no sort of mechanistic commonality in terms of tying these two traits together. So we wanted to ask how rare is antagonistic pleiotropy, or how frequent it is. And there's one previous study in natural yeast isolates showing that antagonistic pleiotropy is rare. And most other studies of antagonistic pleiotropy have largely measured only beneficial mutations that arise under selection. And so, for example, with Lenski's long-term evolution lines, people have asked, if we look at all of these beneficial mutations that arose in those populations, do they tend to have antagonistic pleiotropy in terms of other resources, other than glucose and so on? And so we have a relatively biased view of what is the incidence of antagonistic pleiotropy with a random set of mutations that are likely to occur in populations. And we understand much more about what happens under specific conditions of strong selection. Okay, so we wanted to test this, and we use mutation accumulation regime. So what's happening here is we put bacteria out on a plate. This is E. coli. And every day we pick a single colony, transfer it, and keep on transferring, right? And we freeze stocks at some time interval. And what we do then is go back to these frozen stocks and estimate, given the known mutation rate for this wild type E. coli, at what time point would we have stocks that would have, on average, 1 mutation per genome, 2 mutations per genome, 3 mutations per genome, and so on. And then you can measure growth rates of all of these lines from all these different time points on a battery of resources. So all these populations evolved in very rich medium. This is LB. And we can ask then, as they evolved under minimum selection, because it's rich medium, because it's severe bottlenecks, right, every few generations, how did their fitness change when they accumulated a random set of mutations? We can then plot for each pair of resources on which we measure fitness, which is measured using this, in this manner. And all of these data points, let's say, are the impact of each replicate point that we have, right? So we have 38 replicate lines times 3 time points, right? And then here in this regime, we can start asking what is the impact of each mutation. So let's say we have some threshold for error that's inherent in our measurement. So because we measure growth rates at these large scale, there's some error, so we exclude these guys. We say, okay, they kind of look like they're different from 0, 0, but we will say that there's no change effectively. And now these populations, these mutants are, the red ones here, are what are synergistic pleiotropic mutations, right? So where they have improved on both resources, which are these guys, or they have become worse on both resources, correct? Similarly, these guys, the black ones, are the ones that are showing antagonistic pleiotropy, where they've gotten better on one, but worse on the other, and vice versa, right? And then the open circles here, the blue ones, are mutants that are better in one resource or have changed in one resource, but have not changed in the other resource. Everybody okay with this? This is just a schematic, yeah. Any questions so far? Okay, great. So basically we do this whole thing for all of each pairwise resources, and then we get distributions of fitness effects, right? So here is fitness on the x-axis, here's the frequency of the total set of mutants that we have. How many, for example, have improved fitness in trehalose, one of the sugars we tested? There's just this tiny fraction here. Most of the mutations are neutral or nearly so, and there's a whole bunch of deleterious mutations here, right? And so similarly for glucose and so on and so forth. Now what we want is to also be able to say, well, what is the expected null distribution of antagonistic pleiotropy and synergistic pleiotropy and so on, right? If we didn't have this particular set of mutations that we got. So what we do then is we generate a null distribution using these DFEs that we have. So we draw randomly from each pairwise comparison of resources, and we generate a new sort of random data set, right? Because what we're doing essentially here is we're dissociating the fitness effect of a single mutation on these 2 resources which actually occurred, and we're removing, stripping it of that connotation and saying we just randomly pick one mutation. It may have this, it may fall anywhere within this distribution in terms of its effect on glucose. So then we generate a null distribution like this, repeat that a bunch of times, and we get some sort of distribution of what is the probability of antagonistic pleiotropy in such null distributions, right, the 1,000 null distributions we get, and where does our observed probability of antagonistic pleiotropy lie? Yes? Correct, yeah. But, yeah, exactly, yeah. So then we can compare the observation with the sort of expectation if we are removing pleiotropy and asking how likely are we to see what we actually observe just by chance, right? Just drawing randomly from the distributions themselves. And so just to show you, this is what the distribution of fitness effects of these mutations look like for all the resources we've measured. Just to remind you, we have 12 resources, 38 replicate lines, 3 time points, so about 114 data points per resource. I think these graphs are actually older, so there's fewer points here, but anyway, they don't look too much different. But the other point I want to highlight is if I draw a simple line at, this red line is at 0, which suggests that these mutations are neutral. If you think back to what I said earlier, where most of the information we have about sort of fitness effects of mutations comes from beneficial mutations, right? Which is a very small fraction of all the mutations that actually occur. So if you want to understand more generally what happens or what could happen in regimes which are not very strong selection regimes, we want to focus on also what happens on the left side of these red lines. Yeah? Okay. So here's sort of a general picture of what we see. So it's a stack.
I guess I'm
yeah.
a little confused by the null model. If you take given mutations,
yeah.
and you assume that the distribution of the effects of that are drawn from what you see with the one resource,
yeah.
and drawn with other resource,
yeah.
which is scatter plots,
yeah.
all that.
Yeah.
So it's not a direct measure of interaction.
We remove interaction, exactly, right? So we're saying, okay, we have one, in reality, one mutation has all of these potential effects on whatever, right? Other resources. But what we're saying is, if we independently draw mutations from each of these DFEs, what do we expect to find, right? Okay, so if you look at the stack bar plot, I think one of the first things you'll see is that the degree of antagonistic pleiotropy, which is this middle gray bar here, is rather small. You see a lot of synergistic pleiotropy. You also see some mutations where there's no changes. But overall, the antagonistic pleiotropy is very low. Okay? And that, again, sort of goes back to what we'd shown earlier with the beetles, not exactly parallel, of course, but saying that AP is not very, very frequent. And compared to now our null expectations, also antagonistic pleiotropy. Right there. Somebody has a mic? So for example, you find that for each of the resource pairs now, if we ask how often do we see antagonistic pleiotropy versus what we expect from the null, and we table everything up, of total 55 pairwise comparisons for all the resources, 17 cases are where we observe more antagonistic pleiotropy than expected, 1 case where it's exactly equal, and 37, which is the overwhelming number, where antagonistic pleiotropy is rarer than expected. Right? So again, all of this suggests that across different resources and across pairwise comparisons, antagonistic pleiotropy is not very frequent. Okay, we could then also use these data to test another hypothesis which has been around in literature for a while, which is that antagonistic pleiotropy is more likely in large effect mutations. Right? So if you have mutations which have huge effects, whether they're beneficial or deleterious, maybe you're more likely to find that they also have similarly large effects in some other resource and their opposite effects. Right? And so we could test that in cases where we found enough mutations with significant antagonistic pleiotropy. And what you see here is as a function of if you bin each of the effect sizes into sort of arbitrarily very low, low, medium, and high effect sizes, we see that there is a positive relationship in many of the resources, not all. So the red ones are the ones where we see a significant effect and black are not. And then again, we can compare this to our expected null distribution or result from null, and there we expect a sort of a negative relationship. So essentially what this is saying is then beyond what we expect from the null, it seems to be true that if you have large effect mutations, they are more likely to be antagonistically pleiotropic. And remember, this goes beyond just our simple ability to measure fitness effects, right? Because we already corrected for that. Okay, so what does all of this imply for evolution? Suggests that trade-offs may actually not constrain evolution as much, especially in the short term. the incidence and magnitude of antagonistic pleiotropy is resource dependent. So sorry, what I forgot to point out earlier was also, if you remember that stack bar plot, for each resource, there were different fractions of antagonistic pleiotropy overall, right? Which suggests that perhaps in some resources, if you evolve under strong selection, you are more likely to show trade-offs. And this may make sense if you think of the metabolic networks or the genetic networks that underlie resource use. Right? Something as central as glucose metabolism, for example, may be more likely to lead to antagonistic pleiotropy, and perhaps that makes sense. So maybe trade-offs then could play a bigger role during adaptation to specific resources. And large effect mutations are more likely to show antagonistic pleiotropy. And remember, large effect mutations are also the kinds of mutations that we're most likely to see early during very, very strong selection, right? So these huge beneficial effect mutations that you see early and during adaptation, then sort of this slowing off. Maybe trade-offs are more important early on because of this relationship that we observe. Okay, so then that still leaves a lot of questions. How often are natural populations limited by trade-offs? We still don't really know. We get some hints from the beetle work, but for bacterial populations in wild, we have no idea. So trade-offs are thought to constrain adaptation because essentially if you have antagonistic pleiotropy and you've evolved under selection in one habitat, it constrains your adaptation to another habitat because the mutations that are beneficial in this habitat are not beneficial in the other habitat, right? And that typically, for example, is invoked to explain why specialists and generalists both may coexist if there's habitat fluctuations, right? Yeah, so limiting in the sense of, yes, not being able to easily switch because of these trade-offs. We also don't know how does environmental variation alter trade-offs. And we don't fully understand whether pleiotropy can then help explain the evolution and maintenance of generalists as well as specialists in habitats. So I'd like to end there. All of the beetle work was done actually with a huge team of people, summer interns included, but these are the main people who did a lot of the work. Mridula has largely single-handedly done all of the antagonistic pleiotropy work with E. coli. She has a poster about this as well with more details, so please go see the poster. I'd like to also acknowledge Olivier for pointing out the need for a null comparison when I talked to him about some of our early results. This is my lab, and this picture really is just because it just came in from one of my students, and it's really cool. So this is, the blue cells are marking nucleus of a butterfly ovary, and the red cells are Wolbachia bacteria, which are intracellular, and they sort of live inside this ovaries. And they seem to be doing something to the sex ratio of the butterfly. And so I'm just excited because this is a legit looking molecular cell biology kind of image. So I have one in my talks now, even though it makes no sense. Oh, you thought it was a correlation? Yeah. Yeah. No, I'm just so happy that my lab has this now. So yeah. Anyway, so not related to anything that I said before. Thanks so much, and I'm happy to take questions.
So in the E. coli, are they all the different sources that you're giving them? Ones that they would, or close to ones they would see naturally? So, okay, they're probably reasonably well adapted.
Yeah, so I guess most of the sources I showed you at least are all things that E. coli can grow on. Yeah. So if that's any indicator that reasonably well, not great in all of them. So we actually also tested other resources which I didn't include here, where they're just terrible at. And in some cases, some of the mutation accumulation lines, for example, have evolved the ability to use some of these resources, which normally E. coli doesn't. Yeah, those were excluded.
Yes, so can you really say that AP is rare because you can have some other traits that mask these, right? So imagine if you take reptiles that matter, if you have some reptile that's specialist at some temperature regime, and you take it to another temperature regime, it might just change its behavior to adapt to that trait. You can have some other trait that's just masking this instead of,
So you're right, that may very well be something that happens in beetles, for example. But in the bacterial situation, it's unlikely because what we're measuring is all growth rates under very uniform conditions in some sense in the lab. So yeah, yeah. 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 Does that answer your question?
Yeah.
but I mean, in beetles it's true, but also it's unlikely to be true for things like starvation resistance and, which are unlikely to be behaviorally regulated. I can understand resource
Behavior is
choice,
just one example, but
cannibalism,
there could be other traits as well. We
yeah.
Have measured.
No, and that may be what's going on, right? Like that they haven't accumulated enough mutations yet that are deleterious in other situations. And maybe the buffering is essentially provided by redundancy or something else. Absolutely.
Maybe? When I think of antagonistic pleiotropy, the first place I ever heard about it was in life history evolution theory, where people proposed it as one of the possible ways where senescence can evolve because of antagonistic pleiotropy between things acting early and late. And I know that some of the early experiments, Rose and people, looked like it showed antagonistic pleiotropy between early fertility in female Drosophila and long-term survival. But I heard a talk recently by one of Rose's former postdocs talking about those same lines, and over time it looks like that's not true, that they actually, the females that had high fertility eventually also had they were just better. They were just better. So it's interesting that some of the classic examples of antagonistic pleiotropy look more like your beetles. If they're good, they're good.
Yep.
And so that's not really a question. It's more like another data point for you. I don't know.
Yeah. No, so we have actually, lots of bits and pieces that tie together really well with this. So we've measured, for instance, for other projects, immune function and different kinds of immune function in sort of different beetle populations. And again, expecting to find, okay, if you're investing more in external immunity, maybe you invest less in internal immunity, males, females. We don't see very pervasive trade-offs there either. Another thing that comes to mind is we did some recent work on understanding the fitness effects of group sex ratio. And what we found, which was surprising, was that in female biased sex ratios, female fitness was lower. And again, we thought, okay, well, maybe it's lower in the sort of snapshot measurement of fecundity that we were doing initially. Maybe they live longer and so on. Nope. In male biased groups, females just did better at everything, right? And again, that seemed to suggest that, yeah, better is better. And it's very, perhaps plastic also, because these beetles were all from one population and they just sort of went into male biased or female biased groups and consistently showed these sort of, doesn't matter what you measure, they're all better. And so I'm very surprised by that. I don't quite know what to make of it in light of the lack of trade-offs kind of thing. Because I mean, I do believe that trade-offs exist at some sort of higher, phenotypic level, but yeah. No, anybody else?
Yeah, just coming back to the call out the trade-off. So from these experiments, Sherlock, and there, if you want to look at the first mutations that come in serial growth, where they're going from fermentation separation, complicated things, and there are enormous. They're associated with the other things that are going on isn't yet. So my sort of instinct is that there's going to be much more. Much more trade-offs if one starts looking at what they do, how well they survive coming out of the phase, how they switch off.
No, there probably will be.
Presumably something that you can look at.
No, so that's the other thing, right? At what level do you measure trade-offs? So we stuck to sort of growth on basic sugars, simply because we felt that was fairly comparable. And, there was expectation that there would be some connection to, for example, how related the metabolic pathways are and so on. But yes, I'm sure if we go to something like temperature dependent, something else, right? Starvation, stationary phase performance, we might find something different.
But if one looks at certainly the sort of natural environment, probably growing rather slowly. So the rapid growth, relevant. I mean, no one has the ecology we call that. And it's how sort of important rapid growth.
Well, so we do think it is important for some species, and I'll tell you why. So there's a strong correlation between the number of rRNA operons in cells, in genomes, and how fast they are able to grow under whatever conditions, right? That's a very strong correlation. And there's a significant number of bacteria that do show very high numbers of rRNA copies, right, in genomes. And so, and also, by the way, it's clear from experiments that if you delete rRNA operons, you aren't able to grow as fast. So at least to some extent, they are presumably under selection to be growing fast. Are all bacteria under selection? Absolutely not.
But they grow fast and they have to survive well.
Yeah.
But they're not growing.
Yeah.
So it's just, it is a that sort of trade-off. Well, maybe that's it.
Yeah. Likely, we just haven't.
Or E. coli was switching from one producer to another when environment got changed.
Mm-hmm.
It may be somewhat for this niche sort of make it focus on that.
What will work? You mean mutations in genes that would be beneficial for that function versus other? That's essentially what we're measuring with the bacterial work, right? So it's all the same genome that's just getting different sets of mutations. So, yeah. Okay, well, thank you very much.